In order to study action potential and electrical properties of the membrane, we have to define few terms. We already know charge and electrical charge. How would you define it? Is the physical property of matter that causes it to experience a force when placed in an electromagnetic field. And we know there are two types of electrical charges, positive and negative charges. They are positive carried by protons and negative charge carried by electron. And any object with an absence of net charge is referred to as neutral. So if we have to define charge itself, so we can say any substance, any particle that is influenced by uh, an electromagnetic field, positive or negative, is called a charged particle. And we know the SI unit for the uh, measurement of charge is coulomb. And there are certain other units like Faraday, ampere in hour. So if we define current, current is passage of charges through a wire or through an area of wire per unit time is called as a current. So the current is measured in uh, amperes, and it is the number of charges that moves through the wires through a wire per second. Then we have to define potential difference. Potential difference is defined as difference of electrical potential between 2 points, and electrical potential is the amount of energy needed to move a unit of charge from a reference point to a specific point. The potential difference is measured in volts, and in case of cell membrane it is measured, because these are small quantities, so it is measured in millivolts. Similarly, for current, the electrical the SI unit for the unit for electric current on membrane is milliampere, because the current are in smaller quantities on membrane. Then we know there are conductors. Conductor is any substance that can conduct electrical potential or electric current, and this conductor we know have conductivity. That conductivity is the ability of matter to conduct the electric current, and we know there is no ideal conductor, and every conductor pose a resistance when electrical current pass through them. So the conductor with larger cross-sectional area, mean thickness, wire thickness, they have less resistance and more conductance as compared to the wires with uh, less cross-sectional area, or the wires that are thin. And the resistor is a substance that resists passage of charges or current through them, and they are used to reduce current flow uh, in a circuit. And the resistivity is the property of a resistor to resist the flow of charges or current. We know the flow of charges is current. And the resistance and conductance, they are related to the cross section area, they are related to the temperature and to the time. And if temperature is more, resistance would be more and conductance would be less. We know the materials that have more free electrons are good conductor, because the flow of electron in an electrical circuit is actually current. So the metals that have, or the metals that have free electrons, they are good conductor, and those metal that have less free electron, they are good resistor. And then there are material that are called insulator. They does not allow the flow of electron freely between atoms, and the electrons are tightly bound, therefore they are completely inhibiting the flow of charges, so they are called as insulator. Like an ideal conductor, ideal insulator is also non-existent, because there are always free charges. So the insulator, they have more resistance than conductor and semiconductors, but uh, there is no ideal insulator. So the conductivity, the resistivity, and the property of insulation, they are also dependent on the potential difference that is applied across the wire. If potential difference or voltage are more, then conductivity will increase, and if potential difference is less, the voltage are less, then the uh, conductivity will be reduced. We can understand potential difference like that happen in diffusion. The potential difference is, for example, difference of charges between the 2 points. So the current or the charges, they flow from higher concentration to lower concentration. If that difference or potential difference is more, then the tendency of charges to flow toward low concentration would be more. Then there is a term that is capacitor. That is a device used to store an electric charge, and they are made up of one or more pair of conductors that are separated by insulators. So the charges, they are stored on those conductors, and insulator does not allow the passage of charges between two conductors. So the membrane has all these properties, that they have charges on them. The most of the membranes, the membranes of epithelium, membranes of the nerve cells, and the membranes of the muscle cells, they have charge separation on them. Mean the cells, they have charge difference inside the cell, in cytoplasm, and in the extracellular fluid there is difference of charges. So these charges, they tend to flow out of the membrane or from outside into the membrane, and we know the flow of charges is called as current. So there is a current on membranes. And then there is a potential difference, because we said there is difference of charges inside the cell and the outside of the cell. So this potential difference is measured in millivolts. The flow of current is measured in milliampere. And then conductor, the membrane has property of conductance, mean it can conduct charges, because and then the extracellular material, that is a fluid, and intracellular material, that is cytoplasm, again is a fluid. These are working as two conductors, so charges can flow in them. But the membrane itself, because it is made up of lipid bilayer, and we know the lipid layers, they are non-conductors, so we can say they are not good conductor. They work as insulator. They does not allow the charges to easily pass through lipid bilayer. So these two conductors, that is the intracellular fluid, cytoplasm, and extracellular fluid, they are separated by cell membrane. So this whole assembly is working as a capacitor. So charges, they are stored on the membrane inside and outside, and then the membrane, it allows passage of material, but that passage of material is with resistance, that is passage of charges is with resistance. So the membrane also has property of resistance. So if we see our cell membrane, as we said, there are four types of tissues. We know the epithelial tissue, the connective tissues, muscles, and nerves. These are the four basic tissues. And out of these four tissues, three have ion distribution along them. I mean, difference of ion concentration inside of cell and outside of the cell. And we know the uh, extracellular material of the cell is fluid, and intracellular material is also fluid, that is in the form of cytoplasm. So they have difference of charges between them. For example, along an epithelial cell, muscle cell, or nerve cells, the cations that are present, sodium ions, they are 145 millimole in extracellular material, but they are 12 millimole in intracellular fluid, or that is the cytoplasm. Potassium, they are 4.5 millimole in extracellular fluid and 155 millimole inside of the cytoplasm. And then calcium, they are more in extracellular material than the intracellular material. They are 2.5 millimole in extracellular, but in intracellular it is 100 nanomole. It means it is many times less in intracellular material. The anion that are present along membrane are 132 millimole chloride in extracellular material and only 4 millimole in intracellular material. The bicarbonate ions, they are 22 millimole in extracellular material, and they are less, they are about 8 millimole in intracellular material. And then the other negative charges like protein and the phosphate and the DNA, RNA containing phosphate. We know the proteins, they have amino acids that have the uh, side chains that are negative, and moreover they have like sulfate, and the DNA and RNA, we saw they are made up of nucleotide, and nucleotide have phosphate in them. They are negatively charged. So these negatively charged cumulatively, they are their DNA, RNA, and protein, they are not present in extracellular material. They are only present inside the cell. So they are 150 millimole inside the cell. If we see the pattern of these charges, if we see these cation, they are about equal inside and outside of the cell. There is only a difference of calcium ions. The sodium and potassium ion, they are because they both are having single charge, mean they are monovalent. So these two, they are generating about equal charges difference along the membrane. Mean if we sum up them, there is no charge difference inside and outside of the cell. It is only due to calcium. Calcium are more outside of the cell, so making outside more positive as compared to inside. Then if we see these negative charges, they are more outside and less inside. but the protein and DNA and RNA, they are only present inside of the cell, making it more negative as compared to outside. So most of the cell, almost all of the cell of the epithelial tissue of the nerves, that are neurons, and the muscle cell, they are negatively charged from inside and they are positively charged from outside, and that is due to only presence of the protein, DNA and RNA inside of the cell, because they are not present outside. And then there is a small difference between the concentration of sodium and potassium. You have to remember, you have to keep it remembered that sodium are always concentrated outside of the cell and potassium are always concentrated inside of the cell. And the calcium, they are always concentrated outside of the cell. So we have to keep these three ions uh, very clear, that they are, sodium are more inside, outside of the cell, and potassium are very much concentrated inside of the cell, and the calcium, they are always outside of the cell. So why they are concentrated like this? Because we know there are sodium potassium pump. You can recall from previous lecture that sodium potassium pump, they conduct uh, 3 sodium outside of the cell and 2 potassium inside of the cell by using ATP. Because it is a pump, it conducts material against concentration gradient. As we said, sodium are more concentrated outside of the cell and potassium are concentrated inside of the cell. Even then, this pump can push sodium outside of the cell against concentration gradient. So this sodium potassium pump, it is using energy and pushing 3 sodium toward outside and against concentration gradient, and 2 potassium inside against concentration gradient of potassium by using ATP, because it is a pump. So this pump is keeping sodium more concentrated outside and potassium more concentrated inside. So there is a difference of concentration of sodium inside and outside. And calcium, because membrane again is semi-permeable, it will not allow passage of calcium into the cell without any requirement. So calcium, if they go inside the cell, they are pushed out and they are not allowed to enter into cell. So calcium are more concentrated outside. Why calcium is more concentrated outside? Because calcium is needed, we saw that the intracellular transport, that is the through the dynein and kinasian protein, it needs calcium. The dynein and kinasian protein need calcium to transport material. And another thing is that the release of substances from a cell, they need, like we saw the exocytosis, it needs calcium to join the synaptobrevin and the T snare and V snare together. And so calcium is only allowed to go into the cell when there is need of exocytosis. So there are various other enzymes and proteins that need calcium as a cofactor. We know the cofactors are the inorganic substances that are needed by enzymes or proteins for their function. And so calcium, if it enters into the cell without need, then there will be activity of those proteins started, there will be exocytosis without need, and then there would be intracellular transport without need. So the calcium is only allowed to go into cell when there is need of exocytosis of any substances or the intracellular transport through dynein and kinasian, or there is need of some activation of protein or enzymes. So Now the proteins and DNA, because they are more concentrated into the cell, so generally a cell is more negative from inside and it is more positive from outside. That is the ions distribution along a cell. If we see the sodium, if it is more concentrated outside, how it and the membrane, it does not allow passage of sodium ions directly to go inside, and it does not allow passage of potassium ion to directly go outside. It will not allow calcium ion to pass through this membrane. But these charges, that is sodium, potassium and calcium, they can cross membrane through specialized proteins that are the channels, and we have studied them already. So the channels, they could be uh, uh, mechanical gated channel, they can be chemical gated channel or voltage gated channel, dependent on the type of or they can be open channel. But for sodium, potassium and calcium, there are no open channels on membrane. Specifically for sodium and calcium, there are no open channel, but there are few open channel for potassium on membrane. So we know the open channels are the protein embedded in membrane from extracellular to the intracellular side, and these protein, they allow passage of material all the time till the life of the channel or the life of the cell. So open channels, they conduct on the basis of the size and the concentration difference, that is the concentration gradient. So the substances that can fit into open channel and they have their concentration gradient, they pass along their concentration gradient. That is the simple diffusion, that is called as in this case is called as facilitated diffusion. So in case of the sodium, potassium, the calcium and potassium, there are there are closed channels. So sodium channels, they uh, and calcium channels, potassium channel, they can be mechanical gated channel, voltage gated channels, or they can be chemical gated channel, dependent on the stimulus required to open these channels. For example, if a stimulus is chemical, then they are called as chemical gated channel. If stimulus is voltage, then they are called as voltage gated channel. And if stimulus is mechanical or physical pressure or touch, then they are called as mechanical gated channel. So the change in concentration along membrane is dependent on these closed channels. We have to generally see the composition of these channels. If we see a sodium channel, the sodium channels are made up of two gates. Here we can see a two-dimensional structure. So this sodium channel is made up of a gate that is called as inactivation gate and another gate that is called as activation gate. So in a resting condition, the activation gates, they are closed, and inactivation gates, they are open. And when this channel has to conduct sodium into the cell, this activation gate has to be open. These activation gates will get open, and how they get open, we will discuss them later on, because they are made up of protein. So they get open and then sodium can come into the cell, because sodium is more concentrated outside, so along its concentration gradient it can come into the cell, flow into the cell until this channel get closed. So for closure, there is another configuration, that this inactivation gate, it get closed. So when the channel has its inactivation gate closed, then it cannot conduct any charges. So these are the three configurations of sodium channel. So sodium channel has two gates, inactivation gate and activation gate. In normal condition, in resting condition of the membrane, the activation gate is closed and sodium is not being conducted into cell. And when the activation gate gets open, there is conductance of sodium into the cell, and then the sufficient sodium when enter into the cell, these inactivation gates they get closed, and so no more sodium could be conducted into the cell. Why these activation and inactivation gates are closed, we know, and get open, we know the inside of the cell is always negative, and is negative in resting condition, so there are negative charges inside these and the activation gate, they are made up of amino acids that are negative, so these negative charges they push this activation gate to keep it closed. And inactivation gate of the sodium channels, they are made up of positive amino acid, mean amino acid that have positive charges on them, so the negative charges in cytoplasm keep them pulled and keep them open. So in resting condition, because membrane is negative from inside, because there are more proteins and DNA, RNA inside and less positive charges inside, so these inside of the membrane is cytoplasm is negative, it keeps open the positive inactivation gate, but it keeps the negative activation gate closed. But when sodium enters into the cell, the inside become positive due to sodium, because it is monovalent positive charge, so sodium accumulates here, and this sodium pushes, when it becomes sufficiently positive here due to sodium, then these charges can push this activation gate that is made up of positive amino acid to become closed, to push it into the opening of the sodium channel. So this is all about sodium channel, we'll discuss them later also. Then if we see a potassium channel, we said there could be open potassium channel, but they are very few. There are more closed potassium channel or gated potassium channel. So potassium is more concentrated inside. On resting potential or resting membrane potential, the inside of the membrane is negative and outside is positive. So potassium uh, is then this gate, the channel sodium potassium channel, it is made up of only one gate, that is called as activation gate. There is no inactivation gate like in sodium channel. So this activation gate, it is made up of negative amino acids, and inside of the membrane is negative, so these negative charges they keep this potassium channel closed. So when sodium channels get open, sodium enters into the cell, inside become positive, these positive charges can pull this activation gate to open and potassium can leak out. So this is the configuration of opening of potassium channel. So potassium is leaking out. Why potassium activation gate is open? Because the sodium channels were open, sodium entered into the cell, inside become positive instead of negative, and then these positive sodium ions they pull this activation gate to get open, because this activation gate is made up of negative amino acid, so it gets open. Then we have to see what is resting membrane potential. As we already saw that there is difference of charges along the membrane. So first we have to define membrane potential. Membrane potential is separation of charges along the membrane, and the we can define it as a difference in the relative numbers of cations and anions in the intracellular fluid and extracellular fluid. and these potential differences, they are measured in millivolt because they are in minute quantities. And so how we define resting membrane potential? It consists of the we can say a membrane potential present in the cells of the uh, excitable and non-excitable tissues when they are at rest, mean they are not producing electrical signals. What are excitable cells or tissues? Excitable tissues are two, that are the muscles and nerves. Mean they can conduct action potential, and epithelial cells, they are non-excitable, mean they cannot conduct action potential. So resting membrane potential, it exists along three tissue types: epithelial tissue, muscle tissue, and nerve tissue. Two of them, that are the uh, nerve cells and muscle cell, they can conduct action potential, so they are called as excitable. And epithelial tissue, they cannot conduct action potential, so they are called as non-excitable. So but resting membrane potential, it exists along these three membranes, and it is when they are not conducting action potential. So when they are not conducting producing electric signal, when the membrane is at rest. And we know when membrane is at rest, there is contribution of different anions and cations inside the cell and outside the cell for the difference of charges, that is called as potential difference or resting membrane potential. Then what is action potential? Uh, what is the difference of charges between resting membrane potential and action potential? As we said, when there is a resting membrane potential, membrane is negative from inside. But the excitable cells like nerves and muscles, they can have a short term change in this condition, that inside of these cells may become positive when sodium is conducted into the cell, and outside may become negative, relatively negative. So that is called as, and it is spreaded on the whole cell. These changes, they are spreaded on the whole cell membrane of these cells, so they are that is called as an action potential. So action potential is in action potential, the charges, they are inverted as compared to resting membrane potential. So we'll see them later. So resting membrane potential, we said, is the potential or electrical difference in electrical charges inside the cell and outside of the cell when membrane is at rest. So the we know the charges that are contributing. We know there are difference in sodium and potassium. We saw the sodium is more concentrated outside and potassium is more concentrated inside. We saw that calcium is more concentrated outside, making outside more positive. But the real difference is done by the presence of the proteins and DNA, RNA inside the cell, making it more negative from inside. So we have to keep it remember that resting membrane potential, at resting membrane potential, inside of the membrane is more negative and outside is more positive. And we discussed it already that the sodium and potassium, they cannot cross membrane, but they can be transported actively through sodium potassium pump. We know the pump, they push the sodium outside and potassium inside, so making a difference of, because three sodium ions are being pushed outside and two potassium ion inside, so there is a difference of charges. Outside become more positive as compared to inside. And then these charges, they can passively flow through facilitated diffusion, that is through channels, but these channels, they are mostly closed channel. For sodium, they are always closed, and for potassium, there are few open channels. So these closed channel, if they get open, then there could be a change in potential. First we have to define what is electrical signal. The Ion concentration may change across membrane due to change in ion movement across membrane, because we know at resting membrane potential membrane is negative from inside and it is positive from outside. Sodium are more concentrated outside and potassium are more concentrated inside the cell. And when there is change in this concentration, that the sodium goes in or potassium goes out, that change in concentration is called as electrical signal. And why there is change in concentration? We know there are sodium channels and potassium channels, so they get open and change conduct the ions along their concentration gradient, so there is a change in potential across membrane. So why these ion channels get open, that is sodium or potassium channel, it depends on the stimulus that is called as a triggering event. For example, in a neuron, we know the neuron has distinct parts. You know where neurons have three basic parts. We know that is the dendrites, cell body, and axon. So these axon then end at the axon end plate and or synapse. So the there is a difference in structure depending on the location and function of neurons. However, the nucleus and the organelles, they are housed in a cell body from which numerous extensions known as dendrites project like the antenna to increase the surface area for receiving signals. So dendrites, they receive signals, and then the cell body is just to regulate the functions of the we know the cell body has mitochondria, and it has the nucleus that is containing DNA, and for the regulation of the for the production of proteins. These proteins may become channels later on, and then the axon, it is to conduct the signal to the other cells, could be muscles. Then this end of the neuron is called as neuromuscular junction, or it could be another neuron, so that is called as a synapse. So the axons, they are taking signal away from the cell body, dendrites are taking signal towards cell body, and axons are taking signals away from the cell body. The dendrites and cell body, they have the protein receptors for getting signals, because they are receiving signals. So they are the input zone, that is dendrite and the cell body, they are input zone to receive the stimuli. And here first the in dendrites the electrical signal is produced, that is change in resting membrane potential occurs, and that change may produce action potential or may not produce action potential, but electrical signal, that is change in potential across membrane, it occurs along dendrites and cell bodies. Why this happens? Because there are a lot of sodium channels in dendrites. So here we can see, here it is a channel, that is a sodium channel. So these sodium channels, they could be of any type. They could be mechanical, they could be chemical, or they could be electrical type. It depends on the type of stimuli that is coming on these dendrites. So when there is a stimulus, that will open the channels. For example, if it is a chemical sodium channel located here, and these dendrites, they are receiving chemical signal. For example, this neuron is for olfaction, that is the sense of smell. So the chemicals, they evaporate from our chemical and they go into our nasal cavity, and then they bind with dendrites of these neurons. So these dendrites, they have chemical gated sodium channels. So the sodium is concentrated more outside. When these chemical, they will bind with these chemical sodium gated channel, chemical gated channel, then so they get open and sodium will rush into the cell because its concentration gradient is toward inside. And we know there could be two type of chemical gated channel. They could be inotropic, they could be metabotropic. If they are inotropic, they will conduct sodium directly, they will get open directly when a chemical will bind with them. And if they are uh, metabotropic, then they will run a cascade of chemical reactions, and then they will get open the sodium channels, and then sodium will rush into the cell. In any case, the sodium will go into the cell to change the to produce an electrical signal. If it is these neurons are the cells for like receiving sound waves, if it is a cell for sound detection, then the sound waves are uh, a mechanical phenomena. So there would be sodium channels that are mechanical gated channel. So when this mechanical, that is the force of we know in ears, the air waves, they enter into the ear and then they vibrate those bones, three bones, and then in internal ears, and then there is movement of fluid in cochlea. We'll learn it in senses. So the movement of fluid in cochlea uh, moves the cells in that are sensory cells in the cochlea, and those cells, they have sodium gated channel, mechanical gated channel. So those sodium mechanical gated channel got open and sodium enter into those cells to change the potential, that is called as an electrical signal. It could be an electrical gated channel if electrical stimulus is coming on these dendrites. So these dendrites are the receiving portion of the cells, of could be epithelial cells or could be muscle cell. They, if they are receiving mechanical stimuli, then these channel would be sodium mechanical gated channel. If they are receiving chemical stimuli, then these channels would be chemical gated sodium channels. And if they are receiving electrical stimuli, then these channels would be electrical gated channels or voltage gated channels. So the dendrites, they have sodium gated channels. The muscle cells, they also have sodium gated channels. So the uh, this portion of the axon, this portion of the neuron is called as an axon, and it is conducting action potential away from the cell body. So it could be a single elongated tubular extension that conduct action potential away from the cell body and terminate on other cell. These cells could be glands, they could be muscle, they can be the other neurons. So these axon, they can give rise to side branches. These side branches, they are called as collaterals. So these are collaterals, and here it is another collateral, and then within collateral there is another branch, another collateral branch. So this portion of the axon, that is the first portion of the axon that is connected with the cell body, this portion is called as axon hillock. And here the it is also called as trigger zone, because when sodium ions, they enter into the dendrites and cell body, they get accumulate here. And axon has only sodium voltage gated channels and potassium voltage gated channels. we have to keep it remember that axon of the neurons, they have sodium voltage gated channel, they have potassium voltage gated channel, they have calcium gated voltage gated channel. They all are electrical channels, they are not mechanical or chemical. So the axons, they are not receiving portion of the stimuli, they are just propagating, so they are having the voltage gated channel. Dendrites can have mechanical, chemical or electrical gated sodium channels, but and the cell bodies, and the axons, they have only voltage gated channels for sodium, for potassium and for calcium. So the sodium that is entering from dendrites and the cell bodies, they get together here in the portion of axon that is connected with cell body, the first portion. So this portion is called as hillock of the axon, and it is also called as triggering zone, because here the all the charges ions that are entering, they get sum up, and if they are enough to open sodium voltage gated channel in axon, then they will produce action potential, and if they are not sufficient, they will not produce action potential. We'll see it in later section. Same is the case with muscles. We see here an axon is coming, a neuron, and is coming at the muscle membrane, so on plasma lemma, and this plasma lemma, the neurons, they are releasing neurotransmitter, and these neurotransmitter, they are getting attached to sodium chemical gated channel. So most of the muscles, they have sodium chemical gated channel on their membrane. So the muscle membrane, first it was on resting membrane potential, it means the membrane was negative from inside and positive from outside. Sodium were concentrated more outside and potassium were concentrated more inside. But when neurons, they release neurotransmitter, these neurotransmitter go to sodium chemical gated channel. So sodium chemical gated channel get open, sodium will go into the cell, inside will become now positive instead of negative. So this change in potential is triggering event. It is the electrical signal that is produced. If sufficient ions will enter, they can be propagated in the next section. So this section will become first positive, that is called as an electrical signal, or it is an triggering event. So we summarize that either in neurons or muscles or epithelial cells, when there is a triggering event, that will mostly open sodium channels. These sodium channel could be chemical gated channel, they could be mechanical, they could be voltage channel, depending on the type of stimulus that is coming onto these cells. So there are two or three types of mainly electrical signals. They are called as graded potential or local potential, and mainly the action potential. So graded potential is, we know the dendrites, they are, for example, for a neuron, the dendrites, they have sodium channels, they are input of the neuron. From here stimulus will change the potential, produce signal. So for example, if this stimulus was chemical stimulus, then sodium chemical gated channel in dendrites, they will get open, sodium will go into, sodium ions will enter into the cell, inside was negative, now it will become positive. These positive charges, they will flow from here, would be the cytoplasm field, so they are passing through this cytoplasm, they are going to this region of the axon that is called as axon hillock. In this region there is initiation of action potential, because here are sodium voltage gated channel. If these charges, they are insufficient, they cannot open sodium voltage gated channel here, then these sodium channels, they will diffuse off, because they will go, they are not opening sodium voltage channel here. So these sodium voltage channels, they will be they will remain closed. More sodium cannot enter into the cell. These sodium that have entered through dendrites, they are passed this area, that is space, and they are past time. So they will go to this region, they will diffuse into cytoplasm because cytoplasm is a watery medium. They will go into the next region and they will die off. For example, if 5 ions have entered and they have reached only 3 here because the cytoplasm has diffused few of the ions in it, because it is water, so ions will get lost in this water. And 3 have reached here. Actually 5 was required to open sodium channel here, but these 3 they cannot open sodium electrical channels. So these 3 they will go to next region, and these 2 of them, for example, when they will travel equal distance, they will get lost, and only one can reach in next region, that is again insufficient to open sodium voltage channel. It will get lost in a little distance, so it cannot travel on the whole neuron. This change in potential cannot travel on the whole neuron, and because the insufficient ions enter into the neuron through the dendrites. Why insufficient ions entered? Because the stimulus was not sufficient. So that potential is called as graded potential. It is called as degenerative potential. It is also called as degenerative because it is degenerating along the length of the membrane. Or it is called as local potential because it is in only a local area. It is not passing on the whole membrane. It is remaining on the local area. So if these here enters 8 ions, 5 are required here, and 8 ions have entered, then these 8s, they are, for example, 2 are entering from here, 2 are entering from here, 2 enter from this dendrite, 2 from here, they will get sum up here. And for example, 3 of them have lost, or 2 of them, for example, have lost, and then 6 are reaching here or 5 are reaching. They are sufficient to open sodium voltage channel here in the axon. So if they reach here, we know the sodium voltage channel, they have 2 gates, activation and deactivation gate. Activation gate was closed, and the inactivation gate was open. Now these charges, they can open activation gate and sodium will go into the cell. And for example, 3 sodium, when for example if 6 have reached here, then the 2 sodium, when they will enter here, they will again become 8, and they will move equal distance. When they will move equal distance, they again 2 of them will get lost, and 6 will reach in the next segment. And again here the sodium voltage channels, they will get open, so 2 sodium will enter again and they will become 8 again. 6 plus 2, they will become 8, and then they will go in equal, they will travel equal distance and go to the next segment, and they 2 of them will get diffused and only 6 will reach, and these 6 are sufficient to open sodium voltage channel, and 2 sodium ions will enter into the cell, and again they are 8. So they are being propagated. It is regenerated. The potential that the ions that enter from here in dendrites, they are traveling distance, they are getting diffused into the cytoplasm, but the new opening of new channels is regenerating that potential, so it is again regenerated. So it is called as, it will travel on the whole membrane like this. So this potential is called as action potential, or it is called as the regenerative potential. And it is not graded potential, it is of same value, that is traveling from the whole membrane, that is the 8 ions that are traveling through whole membrane. But in last case we saw in graded potential, 5 entered, 2 are diffused, 3 reached here, then two diffuse, one reach there. So that is in graded fashion. It means it is the values of charges are being changed 5, 3, 1. So in case of action potential 8 enter, for example, 6 reach here, again become 8, again reach 6, again become 8. So it is a same potential that is traveling on the whole membrane. So action potential is not graded potential, it is the regenerative potential. It is action potential that is traveling on the whole membrane. It is a change that is traveling on the whole membrane of neuron or muscle, and it is only occurring on neuron and muscle, these tissues that are called as excitable cells. The action potential is not occurring on epithelial cell or any other type of cell. So graded potential, it happens on neuron muscles, and they are just to the they will be localized in an area. It is degenerative with time and space, and it is graded. So what is generator potential? Actually, in epithelial cells, action potential is not running. Epithelial cells are mostly, they are linked to, on their base there is a neuron. So the epithelial cell, they are acting as a receptor. They have resting membrane potential, and then there will be a stimulus that will open channels on the epithelial cells, and there would be change in potential of epithelial cells that would be conducted directly to neurons on the dendrites of the neurons. So that change in potential in epithelial cell is called as generator potential. In this diagram, we can see that sodium ions are entering from different dendrites into the axon, into the neuron, and these entry of these sodium ions, they are getting sum up into axon hillock. So in axon hillock there is two type of summation. That summation is called as spatiotemporal summation. Spatial means with reference to space. It means the ions that are entering into neuron through different dendrites, they get sum up in the axon hillock. And secondly, the sodium ions that are entering first, and then later on they get sum up into the axon hillock. So this is the spatiotemporal summation. So if after summation these ions are sufficient to open sodium voltage channel here, then there will be production of action potential. If these ions are insufficient, they would be diffused, so there would be a graded potential and or degenerated potential. There are few terms that are used to describe the change in potential on the cell membrane. The first one is polarization. Polarization, as the word describes, the charges that are separated across the cell membrane. It means there are formation of poles, that is the difference in charges across the membrane. When there is difference on the of the charges across membrane, then it is called as membrane is called as polarized, and the process is called as polarization. It could be any charge difference other than 0 millivolt. It means there could be positive outside or negative inside, or negative outside, positive inside, anything. There is difference between difference of more than 0 millivolt, that is called as polarization. It means membrane is polarized. Then we have depolarization. So depolarization is when there is decrease in difference between inside and outside, charges between inside and outside of the membrane. If there is change in potential and it is near to zero, when inside and outside has difference of near to 0 millivolt, then it is called as depolarization. So then the repolarization is again if membrane achieves a difference that was present on the resting membrane potential, that is the potential that is greater than a lot of greater than 0 millivolt. For example, in depolarization, if the charges they are near to minus 20 or plus 20 millivolt, and that is called as depolarization if the resting membrane potential is minus 80 millivolt. And hyperpolarization is when the membrane potential was the resting membrane potential was 80 millivolt, but now it become minus 90 millivolt, that is greater than minus 80 millivolt, so it is the change in that is called as hyperpolarization. Mean membrane has become more polarized. The potential difference or charge difference inside and outside has become greater. So these three terms are very important to understand the action potential.